So in my talk this morning, I'm going to present how Edinburgh Instruments can combine spectroscopy and microscopy for high-resolution hyperspectral imaging. A brief outline of what I'm going to talk about over the next 30 minutes or so. I'm going to start with an introduction into what microspectroscopy and hyperspectral imaging are. And I'm then going to move on to imaging with the micro-PL add-on for the FS5 and FLS 1000 photoluminescence spectrometers. And then I'm going to finish off with moving on to imaging with the dedicated RM5 and RMS 1000 imaging microscopes. So to start us off, what is microspectroscopy? So in essence, we want to measure spectra, that could be Raman spectra or photoluminescence spectra, as a function of position on the sample. So to do this, we put the sample on a motorized stage, and then we create a grid, and we record spectra as a function of position on the sample. And then we choose an analysis parameter, and that could be the peak position, or it could be the ratio between two peaks, or the peak intensity. And we use that chosen analysis parameter to color code our image, shown in the bottom right. And that creates a hyperspectral image of the sample. So some examples of hyperspectral images are shown here on the left. So the first one is a 2D of a plant cell, but we can also do hyperspectral imaging in 3D, so we can measure in the Z dimension as well. And the key thing here is in hyperspectral imaging, every point in these images has a data set behind it. It has a spectrum associated with it. And we can do hyperspectral imaging both from steady-state spectral measurements, but also for lifetime imaging using Edinburgh Instruments, MCS and TCSPC electronics to measure fluorescence and phosphorescence. At Edinburgh Instruments, we have 3 imaging platforms. We have the micro-PL, which couples to the FS5 and FLS 1000. And we have the dedicated RMS 1000 and RM5. Below each of these imaging platforms is the different techniques that these can measure. And throughout the rest of my talk, I'm going to introduce these techniques one by one and explain the type of information that they can give you. And to start off, we'll start with the micro-PL add-on. So as I'm sure many of you are aware, the micro-PL is an add-on that couples to the FS5 or FLS 1000. And this is a really powerful combination as you get all the spectroscopy benefits of the FS5 and FLS spectrometers, but now you can also do hyperspectral imaging with them and microspectroscopy. And so with the micro-PL, you can excite your sample with either the xenon or laser excitation and measure single-point spectra and lifetimes at different positions on the sample, and also FLIM and PLIM photoluminescence imaging. An example of how this would couple to an FLS 1000 is shown here. So instead of the sample module, we have a liquid light guide coupling. So this takes our excitation light into the microscope, we excite our sample, and then we collect our emission light and send it back to the spectrometer to detect on the PMT. And now is just a photograph of what this looks like in practice. At the bottom of the image is the excitation coupling for the xenon lamp. So if you want to excite your sample using the xenon lamp of the FS5 or FLS, And then above that is the laser coupling for point excitation using the Edinburgh Instruments laser sources. And there's four main measurement types we can measure using the micro-PL. So they're wide field imaging, point excitation, lifetime imaging, and transmittance imaging. And I'm now going to discuss the first three of these one by one in my talk. So we'll start off with wide field imaging. So in wide field imaging, we're using the xenon lamp, in this case of the FS5, and then we're sending that to the microscope to excite the sample. And then we're detecting the emission using the CMOS camera of the microscope. And this is an example of wide field imaging of some phosphor nanoparticles. So we can see here we've got two different types of nanoparticles. One emits orange and one emits green. And the key point for this type of imaging is this is not hyperspectral. This is just classic fluorescence imaging using the CMOS camera of the microscope. Instead of sending the light to the camera, we can instead send it back to the FS5 and detect using the PMT detector. And if you did that, you would get the total spectrum of the orange and the green detected using the PMT. So with the xenon lamp, we can get the total spectrum of our field of view, but what if we want to measure the spectrum of the individual particles? So to do that, we change to a laser source. So now we're not using the xenon lamp, we're exciting using the laser source, and this gives us a much smaller spot size. So we can target individual points on these samples, and then using the FS5, we can measure either the spectrum or the lifetime of the individual particles. And this is just to illustrate the difference between using the xenon excitation and using the laser excitation. So if you use the xenon lamp to excite, the spot size is typically 200 microns to 2 millimeters in size. And you would excite the entire field of view, and you would get the combination of the two particles. If we excite using the laser sources, we can get spot sizes down to 2 microns, and therefore we can target each particle individually. Now moving on to lifetime imaging. So now if we use the motorized stage of the micro-PL, we can create our lifetime map. So we can use the TCSPC or MCS electronics of the FS5 to record fluorescence or phosphorescence decays at each point on our area. And then we can calculate the lifetime and create a color-coded image. And this is an example of what this would look like for this sample. So on the left, we have the intensity image that you saw previously, and on the right is now the lifetime image. So we can see that the orange particles are color-coded in red. They have a lifetime of around 700 nanoseconds, whereas the green particle is color-coded in blue and has a shorter lifetime of about 100 nanoseconds. So that's lifetime imaging using the micro-PL. And to do this imaging, we have a dedicated software called FluoricoMap. So FluoricoMap runs alongside the Fluorico software that you'll all be familiar with that controls the instrument. So FluoricoMap is our dedicated microscopy software, and then we use Fluorico for the spectroscopy. I'm now just going to present a couple more examples of where lifetime imaging can be used on the micro-PL. So the first example I have is an aluminium indium gallium phosphide LED chip. So this is just a standard microscope image, just in bright field mode. But if we use the EPL laser source to excite the sample, we can measure this lifetime image. So we can see that the area between the LED chips is black, so there's no emission here. And then on the LED chip itself is color-coded in red. And so this has a lifetime of around 11.3 nanoseconds. However, we can also see, as we move towards the edges of the chip, that the color's now turning to orange. And at the edge of the chips, the lifetime is shorter. It's around 9 nanoseconds. So using the micro-PL, we can explore how the lifetime varies over the sample. This is the same LED chip, but now we've acquired a higher resolution image. So we've now switched to a 40x magnification objective to focus in on one area. And we can see a similar effect of the area in the center of the chip is color-coded in green, which has a lifetime of about 5.5 nanoseconds, whereas the edge of the chip has a shorter lifetime, around 3 nanoseconds. Moving away from semiconductors towards biology, the micro-PL can also be used to analyze biological samples. So this is an example of a convallaria root that's been stained with a fluorescent dye that's sensitive to the microenvironment of the root. And the top is a standard microscopy image, and then the bottom was acquired on the micro-PL. And we can see that the lifetime varies depending on the microenvironment within the root cell. And so we're changing from about 0.9 nanoseconds to around 3 nanoseconds lifetime throughout the cell. And just a final example before I move on to the next section is using the micro-PL to investigate solar cell materials. So this is an example of a perovskite solar cell stack where we have vertically aligned carbon nanotube towers as the whole extraction layer. So these carbon nanotubes are deposited onto the ITO, and then we measure a film image of our perovskite surface, and we can see that the charge carrier lifetime on top of the carbon nanotubes is shorter. So these dark squares indicate a shorter lifetime, and this indicates we're getting more efficient extraction of holes into the carbon nanotube whole extraction layer. And then we can just see an example, 2 decays from 0.1 and 0.2, showing the shorter lifetime in region 2. So that was the micro-PL system. I'm now going to move on to the dedicated microscopy systems, the RM5 and the RMS 1000. And the key difference between these and the micro-PL is that these are CCD-based microscopes. So we have a spectrograph with a high-efficiency CCD camera, and using this combination, we can capture the entire spectrum in a single shot. And then the sample is again placed on a motorized stage, and we can capture either Raman or photoluminescence spectra as a function of position on the sample. The first type of imaging on the RM5 and the RMS that I'm going to discuss is Raman imaging. And we just heard a lot about Raman in the previous talk, so I'll go through this slide quickly. But in essence, it's an inelastic scattering technique that provides chemical information about your sample. So it essentially provides a chemical fingerprint as different functional groups or different phonon modes give a unique Raman spectrum. And here's an example of why this is such a useful technique. So here we have a pharmaceutical tablet, and we're actually looking here in this dip in the middle of the tablet, and then we've imaged across that using Raman. And this tablet contains 3 different drugs, paracetamol, aspirin, and caffeine, and each of these has a unique Raman spectrum, and therefore we can create this chemical image of the tablet surface using Raman. We can see the distribution of the different drugs. Raman imaging is also extensively used in material science. On the left here is an example of a silicon semiconductor wafer that's been imaged using Raman, and this silicon wafer contains a defect. So we can see here the conventional white light microscopy image, and then this image on the right is looking at the position of the silicon phonon mode. The red region is the standard position of the phonon mode, around 520 wave numbers. But as we move towards the defect at the top, we can see that the position of the phonon mode changes due to the disorder introduced by the defect. Instead of mapping the peak position, we can also map the full width half maximum of the phonon mode. And again, we can see that as we approach the defect, that the full width half maximum of the mode broadens due to the increased disorder in the silicon lattice. On the right is a different materials example, now using on graphene. So graphene is a very important 2D material for electronics, but its properties are very sensitive to layer number. So here we've used the Raman imaging to determine the number of layers in the graphene. So graphene has 2 different Raman peaks, and by looking at the ratio of these 2 peaks, we can determine which regions are monolayer and which are multi-layer graphene. I'm just going to run through a couple more examples of Raman quickly. So it's also used extensively in forensics. So this is a fingerprint that had paracetamol on it from your thumb, and then the paracetamol is detected, which is shown in the red regions. Raman is also highly sensitive to polymers and plastics, and a very important environmental concern at the moment is microplastics. And using Raman imaging, we can identify the location of the microplastics, and from their Raman spectra, we can identify the type of plastic. A couple more examples to finish off the Raman section. So for geological analysis, we can probe through the different layers in the mineral and identify their chemical composition. And for art and heritage and looking at historical artifacts, we can use the Raman to analyze the different pigments within the historical artifact to determine when it was made. So that was the Raman imaging, but the RM5 and the RMS1000 can do more than just Raman. They can also measure photoluminescence. And in fact, we can combine the 2 together. So this is an example of Raman and photoluminescence imaging of a gallium nitride chip, and we're exciting it using a 325 nanometer laser. So first, we acquire the Raman spectrum from the chip, and we're analysing this region up in the top left, marked in the green box, and then this image on the right is the phonon mode of the gallium nitride. And so that is plotting its intensity. So that top right image is essentially showing how thick the gallium nitride layer is. Then on the bottom left, we're now monitoring the position of the phonon mode, and that tells us what strain and stress are within the gallium nitride. So you can see that there's this region in red, which has its phonon mode shifted to higher wave numbers, and that tells us that the gallium nitride is under compressive strain in that region, which has shifted the phonon mode. And then if we switch over to photoluminescence imaging, we can get additional information. So now we're monitoring the two photoluminescence peaks of the gallium nitride. There's a peak in the UV around 3.4 electron volts, and an additional peak at 2.38 electron volts, which is from indium gallium nitride. And then we're creating photoluminescence images of those two peaks. And we can see that the 2.3 electron volt is only emitted from a distinct region in the sample. And this is actually because the gallium nitride is thinner here, and then we can see the indium gallium nitride layer underneath. And so using a combination of Raman and photoluminescence, we can get additional information. So that was Raman and photoluminescence spectra, but we can also add photoluminescence lifetime. This is an example of cadmium selenide quantum dots that have been dispersed in a glass matrix. And we can image using Raman, photoluminescence, and lifetime. So in the Raman, you can see the phonon modes of the cadmium selenide quantum dots, and also the glass. Then in the photoluminescence spectral image, you can see the location of the quantum dot clusters. And then finally, if we switch over to TCSPC, we can measure the variation and the photoluminescence lifetime of the quantum dots. To wrap up my talk, I'm just going to give a last section, which is in more specialized microscopy techniques that are specific to the RMS 1000, which is nonlinear microscopy. So the first nonlinear microscopy technique that I'm going to talk about is two-photon excitation. So I think everyone in this room will be very familiar with one-photon excitation, where we excite with blue light, excite the sample from the S0 to the S1, and then measure the fluorescence. But instead, we can also use 2 photons of infrared light and then generate 1 photon of green. And the advantage of 2 photon is that the infrared light can typically penetrate deeper into samples, particularly for biological imaging. And also, because it's a non-linear microscopy technique and it scales with the square power of the laser, we only get fluorescence emission from the focal plane. And so it gives you very good Z resolution. And to do this on the RMS 1000, we need quite a high power pulsed laser. So this is typically a picosecond or a femtosecond titanium sapphire or fiber laser. And we can couple these into the RMS 1000. And that's what it looks like in practice. So we couple through this side port. And then we can generate 2-photon fluorescence images. So this is an example of an intestine section that's been stained with a fluorescent dye. And in the green region, we can see the villi of the intestine. And then we can also see we've got an additional signal shown in pink, which is second harmonic generation that I'll touch on in a second. And that shows you the collagen and the wall of the intestine. So that's spectral 2-photon. We can also do 2-photon FLIM using the TCSPC electronics of the RMS 1000. So this is now a FLIM map, and we can see that the fluorescence lifetime changes depending on the metabolic state inside the intestine. So the intestinal crypts around the wall have a shorter fluorescence lifetime than the villi. And then the very last technique before I wrap up is second harmonic generation. So this is a similar technique to 2-photon fluorescence, except we're not absorbing the light this time. We're converting 2 photons into 1 photon half the wavelength. And this is a very powerful technique as it's sensitive to symmetry. So it only happens in materials with non-central symmetry. And an example of SHG imaging is this tungsten diselenide. And so we can see that in region A, we have an SHG response at 515, which is exactly half the 1030 nanometer laser. In region B, we've got double the intensity of our SHG response. So this shows you that the layer A and layer B are aligned with each other. However, when we get to region C in the middle, you can see that the SHG intensity is reduced, and this tells us that region C is aligned 180 degrees with respect to region A and B, and you get a restoration of the symmetry which quenches the SHG. And the real power of the RMS 1000 is that you don't only do SHG, you can do all the techniques that I discussed throughout the rest of the talk, and you can combine them together to give a complete multimodal characterization of your sample. So final slide now, just to wrap up, and it's really just to point out what the differences between the different imaging platforms are. So you have the micro PL, which couples to your FS5 and FLS 1000. It can do single point spectral and lifetimes, and also FLIM imaging. We then have the RM5, which is CCD based, so that's excellent at Raman and Photoluminescence spectral mapping, but can't do lifetimes. And finally, we have the RMS 1000, which is our deluxe imaging solution that can do all the techniques discussed. Thank you very much for your attention. 谢谢。